Welcome to the deep dive. Today, we're looking at biomolecules.
Yes, specifically diving into the foundational concepts of Chapter 9.
Right, Chapter 9. And we're going to kind of strip away all those complex diagrams for a second. We really need to start at absolute ground zero.
The raw chemical ingredients of life.
Exactly. And, for you listening, I want to set the stage here. I was actually looking out the window earlier today at a rock, and then a dog walked by.
A classic comparison.
Yeah, and I was thinking about this text we're diving into. It is incredibly counterintuitive to think that a rock and a dog share the exact same basic ingredient list.
It really messes with your intuition.
It does, because visually, structurally, experientially, a bird and a rocky cliff face could not be more different.
Not at all.
But the central premise we are tackling today is why a textbook about living, breathing biology starts by analyzing a dead piece of the Earth's crust.
Well, it's the perfect place to start. And frankly, it is the only logical place to start if you want to actually understand biology rather than just, memorize it.
Right.
Welcome to you listening. I really want you to think about why this topic even exists.
Why do we even care about the dirt when we're studying life?
Exactly.
Yeah.
Because for centuries, humanity was trapped by this idea called vitalism.
Vitalism.
Right. We looked at the dog and the rock, and we just assumed the dog must possess some magical, unquantifiable life force.
Like a soul or some mystical living matter.
Exactly. Something that was fundamentally absent from the dirt. We wanted life to be magic.
We really did. It feels better that way.
It does. But what this introductory paradigm does, the very first thing it does, is completely shatter that magical thinking.
It brings us back down to earth, literally.
Down to the dirt. It grounds the entire study of biology in concrete, measurable chemistry. It proves, without a doubt, that there is no magical element.
And that brings us right to the core idea, which, honestly, it still feels like a grand illusion to me.
How so?
Well, you look at the biosphere, right? It's staggeringly diverse.
Oh, absolutely.
You've got towering redwoods, microscopic bacteria, massive elephants. But the text asks a very blunt question for the learner.
Are all living organisms made of the same chemicals, elements, and compounds?
Yes. And the answer it forces us to confront is that chemically, that vibrant forest and that dead rocky cliff are practically identical.
They are twins.
The text says, and I'm quoting here, when we look at a sample of living tissue versus a sample of the Earth's crust, in absolute terms, no such differences could be made out.
That is a massive statement.
It's huge. In absolute terms.
Zero difference.
Zero difference in the underlying inventory. My mind was blown.
It is a massive statement, and it's exactly where that vitalism argument just completely falls apart.
It just disintegrates.
Right. Because when we say absolute terms, we are talking about a strict binary checklist.
Like a simple yes or no.
Exactly.
Mm-hmm.
Is carbon present? Yes.
Right.
Is oxygen present? Yes.
Yeah.
Is iron present? Yes.
Okay, right.
If you run that qualitative checklist on a piece of the Earth's crust, you generate a list of elements.
Yeah, Dan.
If you run that exact same checklist on a human being, a mushroom, or an oak tree, you get the exact same list.
There's no VIP club.
No VIP club of elements reserved only for living organisms. The Earth's crust possesses the entire inventory of elements needed to construct you.
Wow.
I really want you, the listener, to let that sink in for a second. You, with all your thoughts and complexity, are built from the exact same elemental inventory as a handful of gravel.
Which naturally makes me wonder, from a student perspective, how exactly does a lab prove that?
That's the real question.
Right, because we don't just guess that the rock and the dog have the same elements. We don't just look at them and say, yeah, probably the same.
Science requires proof.
Exactly. And the textbook explicitly mentions elemental analysis. But, and this is where I got stuck, how does that actually work in practice? Like, if I take a piece of liver tissue and a piece of granite, what am I physically doing to them in a lab to get this binary checklist without destroying the sample entirely?
Well, you actually hit the nail on the head there when you asked how we do it without destroying the sample.
Do we destroy it?
We absolutely destroy it. Yeah, elemental analysis is a destructive, incredibly rigorous chemical technique.
Okay, so we're not just scanning it with a laser or something.
No. To find out what something is fundamentally made of, you have to break it down to its bare atoms.
Oh.
In a laboratory setting, you might use techniques like combustion analysis.
Combustion, so burning.
Literally burning it. You take a weighed sample of the organic tissue and you burn it in a highly oxygen-rich environment.
So the whole thing just goes up in smoke.
Essentially. The carbon turns into carbon dioxide gas. The hydrogen turns into water vapor.
Okay, so you're turning the tissue into gas.
Right. And then you capture those gases, weigh them, and you can mathematically back calculate exactly how much carbon and hydrogen was in that original sample.
That's brilliant. So we are literally burning the biology away to expose the underlying chemistry.
That is a great way to phrase it, yes.
But I imagine for the inorganic elements, like the minerals, it's a slightly different process because minerals don't just turn into gas.
Exactly right. For things like calcium, magnesium, or potassium, you perform what's called an ash test.
An ash test.
You take that living tissue and you burn it completely until all the organic carbon compounds are oxidized and they evaporate.
The CO2 and water vapor are gone.
Gone. What you're left with is just a tiny pile of ash.
Like what's left in a fireplace.
Very similar. That ash contains all your inorganic elements.
Okay, and then what?
Then you dissolve that ash, usually in an acid, and run it through a mass spectrometer or use atomic absorption spectroscopy.
Those sound expensive.
They are.
Mm.
But basically, these machines shoot light or electrons at the sample to identify the specific elemental signatures.
Okay, so it's cold, calculated physics and chemistry revealing the ultimate truth of the biology.
Exactly. No magic. Just mass and light.
Okay, so the methodology is solid. We burn it, we analyze the gases, we analyze the ash.
Right.
But I noticed something interesting in the text. It's very specific about what biological samples we should test.
Ah, yes. The 3 samples.
Yeah, it specifically says to perform this analysis on a plant tissue, animal tissue, or a microbial paste.
Correct.
Why not just say test a living thing? It feels like those 3 are chosen very deliberately to cover all our bases.
They absolutely are chosen deliberately.
Why those 3?
Think about it from a scientific method perspective. If you are going to establish a universal chemical law for all of life, you cannot just test one tiny corner of the biosphere.
You can't just test human hair and call it a day.
Exactly. Plants represent the autotrophs, right? The producers that turn sunlight into mass.
The base of the food chain.
Yes. Animals represent the complex, multicellular heterotrophs. We consume things.
Right, the consumers.
And then the microbial paste represents the vast microscopic world of single-celled organisms and decomposers.
Oh, that makes total sense. So by testing a plant, an animal, and a microbe, and finding the exact same underlying elemental results.
You are proving that this isn't just a quirk of human biology or tree biology.
It's universal.
It is the common denominator of all life on Earth.
And to make that proof completely bulletproof, we need a control group.
A baseline.
Right, and that's where the Earth's crust comes in. The text uses it as the ultimate example of non-living matter.
I always tell my students, think of the Earth's crust as your chemical baseline.
Your 0 point.
Right. If you want to figure out what makes a living tissue unique, you absolutely must test it against something that is undeniably inanimate.
And dirt is pretty inanimate.
Exactly. The crust is the raw, unorganized stage upon which life evolved.
I love that, the unorganized stage.
By comparing the plant, animal, and microbe to the crust, the authors are forcing you, the learner, to view life not as something dropped down from the heavens, but as something built directly out of the dirt.
So we run the combustion analysis and the ash tests on the living tissues and the crust.
Yeah, they're the data.
We get our results. Both yield a list of elements like carbon, hydrogen, oxygen, nitrogen, so on.
Yep.
And as we established, in absolute terms, the binary checklist, the lists are identical.
Perfectly identical.
But, and this is the huge realization, if the ingredients are exactly the same, the secret to life has to be in the recipe.
Now you're getting it.
Because if I'm an examiner or just a curious student listening to this, I'm thinking, how can we be so different from a Roth if the parts list is exactly the same?
That is the exact paradox the text sets up.
It's a cliffhanger.
It is.
Yeah.
And it provides the answer in the very next breath.
The relative abundance.
Yes. While absolute terms show no difference, a closer examination of the data reveals that the relative abundance of specific elements is what separates life from non-life.
Relative abundance. So it's not about what elements are present.
It is entirely about how much of them are present relative to the total mass.
Okay, let's make this intuitive for everyone listening, because relative abundance can sound like a dry math term.
Give me an analogy.
All right, imagine I go into my kitchen right now. I pull out flour, sugar, water, salt, and yeast.
Your absolute inventory.
Exactly. That is my absolute list of ingredients. Now, if I mix mostly flour and water with just a tiny, pinch of sugar and salt, I bake a dense loaf of bread.
Right, a basic sourdough.
Yeah. But if I take that exact same list of ingredients, flour, sugar, water, salt, yeast,
Same checklist.
same checklist, but I massively increase the relative abundance of the sugar, I just dump bags of sugar in,
Okay?
I'm going to end up baking something completely different, like a really weird sweet cake.
That's a brilliant analogy.
In absolute terms, the bread and the cake have essentially the exact same elemental components, but the relative abundance, the ratio, transforms the final product entirely.
Biology is basically taking the pantry of the Earth's crust and drastically altering the recipe.
Biology is the baker.
Exactly. And the text identifies two very specific VIP elements that get this special baker's treatment.
The big two.
Right. It explicitly notes that the relative abundance of carbon and hydrogen with respect to other elements is higher in any living organism than in the Earth's crust.
Carbon and hydrogen. Not oxygen, not nitrogen, not iron.
Just carbon and hydrogen are explicitly called out in this comparative paradigm.
Okay, so why those two? If nature could have hoarded any element from the crust to build life, why did it choose to massively elevate the relative abundance of carbon?
That's a great question.
Like, what is it about carbon's chemistry that makes it the ultimate biological building block?
It all comes down to elegance and chemical efficiency.
Efficiency.
Right. Carbon is the ultimate team player on the periodic table because of its valency.
It's tetravalency.
Specifically, it's tetravalency, yes.
Yeah.
Carbon has 4 electrons in its outer shell.
Which means it wants 4 more to be stable.
Exactly. So it can form 4 stable covalent bonds simultaneously.
Like having 4 hands to grab onto things?
4 very strong hands. But it's not just the number of bonds, it's the type of bonds. Carbon has an unparalleled ability for catenation.
Catenation. That's its ability to bond with itself, right?
Yes, bonding with itself to form long chains.
So carbon holding hands with carbon, holding hands with carbon.
Precisely. Carbon atoms can bond to other carbon atoms to form incredibly long, stable chains, branch trees, and really complex rings.
It's the ultimate Lego brick.
It is. Imagine trying to build a massive, complex skyscraper.
Okay, I'm imagining it.
You need a structural material that can connect in multiple directions, hold massive weight, and remain totally stable under pressure.
Like steel beams.
Yes, carbon provides that biological scaffolding. Furthermore, the carbon bond is exceptionally strong.
So we don't just melt.
Right, meaning these complex biomolecules don't just fall apart at room temperature, they hold their shape.
But wait, I have a physics question here.
Go for it.
If it's just about having 4 bonds, about being tetravalent, why doesn't silicon do the exact same thing?
Ah, the silicon question.
Because silicon is right below carbon on the periodic table. It's tetravalent too.
It is.
And the Earth's crust is absolutely packed with silicon. It's mostly silicon and oxygen. So why aren't we silicon-based life forms?
That is a phenomenal question, and it really highlights the genius of biological design.
Why did nature pick carbon over silicon?
While silicon is tetravalent, the silicon bond is much weaker than the carbon-carbon bond.
Oh, really?
Yes. If you try to build a long chain of silicon atoms, it becomes highly unstable and incredibly reactive.
So it just breaks apart.
Especially in the presence of water.
In life as we know it.
Happens entirely in water.
Wow.
Carbon chains are incredibly stable in a watery environment. Silicon chains just fall to pieces.
That's a massive difference.
And there's another reason. When carbon oxidizes, what does it form?
Carbon dioxide.
CO2. Right, which is a gas that easily diffuses in and out of cell membranes.
Mm-hmm.
You breathe it out effortlessly.
Okay, and silicon?
When silicon oxidizes, it forms silicon dioxide.
Which is?
Quartz, sand, solid rock. You cannot breathe out a solid rock.
That would be painful.
Extremely. So nature, being the ultimate pragmatist, selected carbon for its structural stability, its versatility, and its compatibility with a watery breathing environment.
That makes so much sense. We literally couldn't function if we were breathing out sand.
Exactly.
Okay, so that explains carbon. But what about hydrogen? Why is hydrogen the other element whose relative abundance is so heavily elevated?
Because hydrogen is the perfect partner for carbon.
The Robin to its Batman.
Exactly. It's tiny, it has a single electron,
Yeah.
and it readily forms very stable bonds with carbon to fill out that tetravalent scaffolding.
So if carbon is the steel beams, hydrogen is like the bolts holding the empty spots together.
Yes, or the exterior panels.
Yeah.
Together, carbon and hydrogen form the hydrocarbon backbone of almost every major biomolecule.
The hydrocarbon backbone?
They are the structural chassis, and upon that chassis, all the functional groups, The oxygen, the nitrogen, the phosphorus are attached to create the actual moving machinery of life.
This is so fascinating, but let's pivot for a second, because from a competitive exam standpoint, from the perspective of a student sitting down for the Neet exam.
Yes, the examiner's perspective.
Right. This distinction between absolute inventory and relative abundance has to be a massive target for examiners.
It is highly weaponized.
If I'm an examiner, my immediate move is to trick a student into thinking life has some magical, unique element.
Or trick them regarding which specific elements are actually elevated.
Right. So as our resident mentor, what do we need to watch out for?
You are thinking exactly like an examiner now. This introductory concept has two classic traps that students fall into year after year. Let's call them the absolute trap and the relative trap.
The absolute trap. Okay, I imagine that looks something like an assertion-reason question.
Yes, exactly.
Something like assertion. Living organisms contain unique, specific elements that are completely absent in the inorganic Earth's crust.
Perfect. And a student who is just relying on their general intuition will read that and think, well, of course I'm made of different stuff than a rock. I'm a living, breathing human.
It sounds so logically true if you don't know the chemistry.
It does. They will mark the assertion as true, and they will immediately lose marks.
-1.
Right. You, as a student listening to this, must vigorously shut down that intuition.
The text is the only truth.
The text is your only truth here. In absolute terms, the lists are identical. There are 0 unique elements.
0.
If an option suggests life possesses an element entirely absent from the crust, it is categorically 100% false.
Okay, absolute trap neutralized. So how does the relative trap work?
The relative trap is trickier because it preys on your broader biological knowledge.
Oh, I hate those. Using your own knowledge against you.
Exactly. If I were studying the paper, I'd probably put nitrogen in a multiple choice question to throw them off.
Okay, let's play it out. How would you ask it?
The examiner will ask, according to elemental analysis, which specific elements have a higher relative abundance in living organisms compared to the Earth's crust?
And the options.
The options will be pairs. Option A, carbon and oxygen. Option B, nitrogen and hydrogen. Option C, carbon and hydrogen. Option D, oxygen and silicon.
Oh, that's evil.
Why is it evil?
Because a smart student, a student who has studied the rest of the syllabus, knows nitrogen is in every single amino acid. It's in every single nucleotide in DNA.
Right.
And they know oxygen is literally what we breathe, and we are mostly water, which is oxygen. They are going to be incredibly tempted to pick an option with nitrogen or oxygen because they sound so profoundly biological.
But here's the catch. The question isn't asking what is biologically important overall.
It's asking about the specific text in Chapter 9.
Exactly. It is asking a very specific question based on the textual distinction highlighted in the introduction. The text specifically and exclusively names carbon and hydrogen as the elements whose relative abundance is distinctly higher when directly compared to the crust.
Just those two.
You must stick strictly to the parameters of the text. Do not overthink it. Do not let your advanced knowledge of DNA distract you from the foundational fact being tested.
So the answer is always carbon and hydrogen in this context.
Carbon and hydrogen.
The precision required there is intense.
That's what separates the top percentiles.
Okay, for you listening, let's lock in the keywords. Elemental analysis is the tool.
Yes.
Earth's crust is the control group.
Correct.
Absolute terms shows identical lists.
0 difference.
And relative abundance highlights carbon and hydrogen.
Perfect.
Okay, we have established this incredible chemical foundation. But as we know, biology doesn't just stop at pure elements.
Life is not just a pile of carbon dust.
Right. These elements have to actually combine.
Yeah.
And this brings us to the actual landscape of the rest of the chapter.
Yeah.
We really need to build a mental map of how we get from these raw, dead elements to a living, breathing cell.
Let's build that map. Understanding the logical flow of this chapter, sections 9.1 through 9.1, is absolutely crucial for attention.
It gives you a scaffold.
Exactly. It gives your brain a scaffold to hang all the complex biochemistry on. It tells a chronological story of increasing complexity.
Let's start with the very first step up from pure elements. Sections 9.1 and 9.2 deal with analyzing chemical composition and identifying metabolites.
Right.
Now, we talked about burning tissue to find elements.
A bust check.
But how do we extract actual molecules? Because if I want to find the sugars or the amino acids in a liver cell, I can't burn it, or I'll just destroy the molecules into CO2.
Right. Combustion analysis is way too destructive for molecular analysis. You need a gentler approach.
So what's Section 9.1 tell us to do?
Section 9.1 introduces the technique for isolating intact compounds. You take a living tissue, say a piece of liver or a plant leaf,
Okay.
and you physically grind it up in a mortar and pestle.
Like making pesto.
Very much like that.
Yeah.
But you grind it with a specific chemical, trichloroacetic acid, or TCA.
Trichloroacetic acid, what does the acid do?
This acid breaks open the cell membranes and dissolves a large portion of the cellular contents without completely destroying the molecules.
Okay, so you've got this acidic tissue smoothie.
Yes, you grind it into a thick slurry, and then you filter it through a cheesecloth or cotton.
And when you filter it, you get two distinct fractions, really.
Precisely. You get the stuff that passes through the filter and the stuff that gets stuck on top.
Okay, break those down for me.
The liquid that passes right through the filter is called the acid-soluble pool.
Soluble because it dissolved in the TCA.
Exactly. This pool contains all the small, low molecular weight compounds. We're talking thousands of different organic compounds, but they're physically small.
And what about the stuff stuck in the cheesecloth?
That is the acid-insoluble fraction. It contains the massive, complex, heavy molecules that precipitate out. They are simply too big to dissolve in the acid or pass through the filter.
This simple physical separation technique, grinding and filtering, is how scientists first began cataloging the inventory of the cell.
It's brilliant in its simplicity.
And that acid-soluble pool, the liquid that passes through, that is where we find the metabolites mentioned in section 9.2.
Yes, the monomers.
The text divides them into primary and secondary metabolites. I think it's really important to understand the functional difference here.
It is a critical distinction for the exam.
Okay, so primary metabolites first.
Primary metabolites are the fundamental molecules that are absolutely essential for the basic survival, growth, and physiological processes of the organism.
Essential for life.
Essential. We're talking about amino acids, simple sugars like glucose, fatty acids, and nitrogenous bases.
So the real basic building blocks.
Right. If an organism lacks primary metabolites, its core metabolism completely halts, and it dies. Every living thing, from a microscopic bacteria to a human being, utilizes primary metabolites.
Okay, so if primary is essential for basic life, what are secondary metabolites? Are they just optional?
Well, optional isn't quite the right word, but they are not involved in primary universal metabolic pathways.
So what do they do?
Secondary metabolites are highly specialized. They are often unique to specific organisms, particularly plants, fungi, and microbes.
We don't really make them.
Not in the same way. They serve ecological functions rather than core cellular functions.
Ecological functions. Give me some examples.
Think of things like natural rubber, essential oils, antibiotics, pigments like anthocyanins in flowers, or toxins like alkaloids.
Oh, like caffeine or nicotine?
Exactly. A tobacco plant doesn't actually need nicotine to run its cellular respiration.
It's not burning nicotine for ATP.
No, but it produces nicotine as a secondary metabolite to paralyze the nervous systems of insects that try to eat it.
It's chemical warfare.
It is. They are the biochemical weapons, the attractants for pollinators, and the specialized tools of the organism.
That is a great way to look at it. Primary metabolites are the basic engine parts. Secondary metabolites are the custom paint job and the defensive armor.
I like that.
But life requires more than just small engine parts, which naturally brings us back to the cheesecloth.
The acid-insoluble fraction.
Right, the stuff that got stuck. The actual giant molecules these elements build, sections 9.3 through 9.6, the biomacromolecules.
Now we are entering the world of giants.
Giants.
Yes. Biomacromolecules have massive molecular weights, usually in the thousands or tens of thousands of daltons.
Daltons being the unit of mass here.
Right. And structurally, they are polymers.
Polymers. Let's use an analogy here.
Imagine a child's box of Lego bricks.
Okay.
The individual bricks are the monomers, the small primary metabolites we found in the acid-soluble pool.
The single pieces.
Right. But when you snap hundreds or thousands of those small bricks together, you build a massive, complex structure. A castle, a spaceship, the polymer, or the biomacromolecule. And the syllabus focuses on three primary categories you must know.
Let's break them down. First up, section 9.4, proteins.
The workforce.
Yeah, if there is a workforce in the cell, it's the proteins.
Proteins are the ultimate biological machines. They do almost everything.
Like what?
They provide structural support, like collagen in your skin and tendons. They act as transporters, like hemoglobin physically carrying oxygen in your blood.
Right.
They defend the body as antibodies. And, as we will discuss later, they act as enzymes.
So what are the Lego bricks for proteins?
The monomers that build proteins are amino acids.
Just amino acids.
Yes. There are 20 standard amino acids, and they link together in incredibly specific sequences to form a polypeptide chain.
But a straight chain of amino acids isn't a functional protein, right? Because Section 9.7 specifically talks about the structure of proteins, and it highlights that the 3D shape is just as important as the sequence itself.
Form dictates function in biology, always.
So how does it go from a chain to a machine?
If you just have a linear string of amino acids, that is what we call the primary structure.
With sequence.
Right, but it doesn't stay that way. Due to chemical interactions between the amino acids, that string immediately begins to coil into alpha helices or fold into beta-pleated sheets.
That's the secondary structure.
Exactly.
Yeah.
Then the entire thing folds in on itself into a highly complex, highly specific 3D globule.
A 3D blob.
Basically.
Yeah.
And that is the tertiary structure. This is usually where it becomes biologically active.
Are there any more levels?
Sometimes. Sometimes multiple folded chains lock together to form a superstructure.
Like Voltron.
Yes. Like hemoglobin, which is made of 4 individual folded chains. That is the quaternary structure.
And the shape is critical, right?
If that 3D shape is deformed even slightly by heat or acid, the protein unravels and becomes completely useless.
It denatures.
Exactly.
Okay, so proteins are the complex machines. Next, we have Section 9.5, polysaccharides.
The complex carbohydrates.
Yes, the sugars.
The monomers here, the Legobricks, are monosaccharides like glucose.
Just single sugar rings.
Right. When you link thousands of glucose molecules together, you get polysaccharides.
What's their main job?
They serve 2 main purposes. First, energy storage.
Like a battery.
Exactly. Plants store massive amounts of glucose as starch. Animals, like us, store it in our liver and muscles as glycogen. It's like a cellular battery pack you can quickly tap into when energy is low.
And the second purpose?
Structural support.
Structure. Like wood.
Precisely like wood. Plant cell walls are made of cellulose.
Which is just glucose.
It is just a different, highly rigid way of linking glucose molecules together. It is actually the most abundant organic compound in the entire biosphere.
That is wild. It's amazing that a potato, which is starch, and a rigid block of wood, which is cellulose, are fundamentally built from the exact same glucose monomers.
It's just how they are linked together.
Just slightly different chemical handshakes. Okay, and the third category, Section 9.6, nucleic acids.
DNA and RNA.
The information vault.
The monomers here are nucleotides.
And nucleotides are a bit more complex than just a sugar, right?
They are. Each nucleotide is made of 3 parts. A sugar, a phosphate group, and a nitrogenous base.
The A, T, C, and G.
Exactly. Nucleic acids store, transmit, and help express the genetic information. DNA holds the master blueprint for every single protein the cell ever needs to build.
Safely locked in the nucleus.
Right. While RNA acts as the messenger, physically carrying a copy of that blueprint out to the cellular factories.
So just to recap, we have the machines, which are proteins. We have the fuel and structure, which are polysaccharides. And we have the blueprints, which are nucleic acids.
That's a great summary.
But having a pile of bricks doesn't give you a house. You need mortar.
You need the glue.
Right. And Section 9.8 is entirely dedicated to the nature of bond-linking monomers in a polymer. We need to know the specific chemical glue for each of these macromolecules.
Absolutely. For Neet, the chemical nature of the handshake between monomers is highly tested.
Okay, let's go through them. Proteins first.
In proteins, the amino acids are linked by a peptide bond.
Peptide bond.
It's a strong covalent bond formed by removing a water molecule, a process called dehydration synthesis.
Okay, water leaves, bond forms, polysaccharides.
In polysaccharides, the sugars are linked by a glycosidic bond.
Glycosidic for glucose. Easy to remember.
Right. And in nucleic acids,
The nucleotides.
they are strung together down the backbone of the DNA strand by a phosphodiester bond.
Phosphodiester, so peptide, glycosidic, and phosphodiester.
Understanding those 3 specific bonds is absolutely non-negotiable for understanding how biological polymers hold their shape.
Okay, we have built an incredible structural map. We have our elements, we extracted our metabolites, we built our massive polymers, and we know exactly how they are glued together.
It's a complete physical inventory.
And this is the big turning point. If we stop right here, we're really just looking at a static snapshot.
A freeze frame.
Right, because a corpse has proteins, glycogen, DNA, and peptide bonds.
It does.
A jar of chemicals on a lab shelf has all of these things, but neither of them is alive.
No.
This transition is what makes sections 9.9, 9.0, and 9.11 so deeply fascinating to me. We finally move from static structure to dynamic motion.
This is where we cross the threshold.
Section 9.9 introduces the dynamic state of body constituents, concept of metabolism.
Yes. The molecules in your body, for those of you listening, are not static fixtures. You are not a statue.
We feel solid.
You feel solid, but chemically, you are a flowing river of reactions. You are ceaselessly being built up and broken down.
So even as we sit here talking,
The glycogen in your liver is constantly being broken into glucose, burned for energy, and simultaneously, new glucose is being stitched back into glycogen elsewhere.
it's chaotic.
It is incredibly busy. This sum total of all chemical reactions occurring within a living organism is what we call metabolism.
And metabolism operates in two opposite directions, right? Anabolism and catabolism.
Correct. Think of anabolic pathways as the builders.
Anabolic builds.
They take simple molecules and consume energy to build complex structures, like taking free amino acids and building a massive muscle protein.
That requires energy input.
Yes, catabolic pathways, on the other hand, are the breakers.
Catabolic crashes.
They take complex molecules and tear them down to release energy,
like digestion.
or breaking down glucose in cellular respiration to create ATP. The constant, beautiful balancing act between building up and tearing down is the actual engine of life.
Which perfectly leads to section 9.11, the living state. How does this metabolism actually define being alive chemically?
It all comes down to thermodynamics.
Physics again.
Always. The text defines the living state as a non-equilibrium steady state to be able to perform work.
A non-equilibrium steady state. That's a mouthful.
It's a profound physical concept. Let's break it down. Think of a battery.
Okay, a standard AA battery.
If a battery reaches total chemical equilibrium, what happens?
It's dead. It goes in the trash.
Exactly. It is completely dead. It cannot power a flashlight. It cannot perform any work. Systems at equilibrium are fundamentally static.
So equilibrium equals death.
Chemically speaking, yes. To stay alive, your body must constantly invest energy to prevent reaching chemical equilibrium.
We have to actively fight it.
Yes. You maintain a steady state.
Mm-hmm.
Your temperature stays around 98.6 degrees. Your blood sugar stays within a tight specific range.
Right.
But you are constantly burning energy to keep those levels stable against the natural forces of entropy.
Wow.
The living state is the active, desperate, beautiful fight against chemical equilibrium.
The active fight against chemical equilibrium. That is an incredible way to phrase it.
It's the truth of biology.
But wait, there is a massive logistical problem here that I just realized.
What is it?
If I mix sugar and oxygen in a jar at room temperature, they will eventually react and break down.
They will.
But it might take years for them to naturally combust.
Centuries even.
But metabolism requires thousands of these complex reactions to happen in fractions of a single second just to keep me breathing.
Millions of reactions.
So how on earth does the cell speed up chemistry so drastically at just 98 degrees?
It simply cannot be done without a catalyst. Which brings us to the grand finale of the entire chapter, section 9.12, enzymes.
Deep enzymes.
Why do you think they ended chapter on biomolecules with enzymes? Because absolutely none of that dynamic metabolism, and therefore no living state, is possible without them.
Enzymes are basically the ultimate biological cheat code.
They truly are.
So how do they work?
Enzymes are highly specialized, incredibly complex proteins. Their 3D tertiary structure, that folded globule we talked about, creates a very specific pocket called an active site.
Like a lock waiting for a key.
Exactly. They act as biological catalysts. In chemistry, for any reaction to occur, the molecules must overcome an initial energy barrier.
The activation energy.
Right. Imagine trying to push a heavy, massive boulder up a steep hill before it can finally roll down the other side.
That initial push is brutal.
That hill is the activation energy. What enzymes do is they fundamentally change the chemical pathway. They essentially dig a tunnel straight through that hill.
So you don't have to push it over the top.
Right. They massively lower the activation energy required for a reaction to proceed.
So they don't change the end result of the reaction. The boulder still ends up at the bottom.
Exactly. They just make it incredibly easy for the reaction to happen at the relatively low temperature of a human body.
How does the active site do that?
An enzyme grabs its specific substrate, the molecule it acts upon, and pulls it into its active site.
The key in the lock.
Right. And once inside, it might physically bend the molecule or alter its internal charge. This makes the chemical bonds highly unstable and ready to snap or form almost instantly.
That's amazing.
A reaction that might take 1,000 years to occur randomly in a jar of water can happen millions of times per second inside an active site.
Millions of times per second.
Without enzymes, the metabolic factory shuts down immediately, the steady state collapses, equilibrium sets in, and the organism dies.
Looking at this roadmap, the logical sequence of this chapter is just breathtaking.
It really is when you step back and look at it.
We start with the stark reality that we share our complete elemental inventory with the Earth's crust.
Out of dirt.
We analyze how carbon and hydrogen ratios elevate us above that dirt.
Relative abundance.
We build those elements into primary metabolites. We string those metabolites into giant biomacromolecules using specific chemical bonds.
The Legos.
We throw those molecules into the chaotic, beautifully regulated, flowing river of metabolism.
Right.
We use that metabolism to fight off equilibrium and achieve the living state, and we coordinate the entire symphony using the hyperfast catalytic power of enzymes.
It is a masterpiece of logical sequencing.
It is the hero's journey of biology, from a single carbon atom to a living, breathing cell.
And if you understand that journey, if you can see that flow, the intricate details of the chapter become fascinating rather than intimidating.
We have covered a massive amount of conceptual ground here today.
We really have.
We've gone from the philosophy of vitalism straight down into the destructive chemistry of elemental analysis, mapped the biomacromolecules, and explored the thermodynamics of the living state.
That's a lot to take in.
It is. So now it is time to synthesize. Let's provide our listeners with some concrete memory anchors.
Absolutely essential.
If you are listening to this right now and you need to lock down the foundational facts to carry with you into your exam, this is the moment to focus. Pause if you need to.
Yes. We are going to distill this down to the purest, most high-yield concepts. Here is what you must remember from this foundation.
Let's do it.
I'm going to lay out the 5 critical facts. Burn these into your memory.
Fact number 1.
Fact number 1. Living matter and non-living matter, specifically represented by the Earth's crust, share the exact same fundamental inventory of elements.
No magic elements.
None.
Yeah.
Fact number 2. Because of this shared inventory, in absolute terms, there is completely zero difference in elemental composition between a human being and a rock. The binary checklist is totally identical.
Absolute terms means identical.
Got it. Fact number 3. The defining chemical difference that actually separates the living from the non-living is relative abundance, not absolute presence.
It's the recipe, not the ingredient list.
Exactly. It is about the ratio, the recipe, the proportions. Fact number 4. The 2 specific elements explicitly noted in the text to have a strictly higher relative abundance in living organisms than in the Earth's crust are carbon and hydrogen.
Carbon and hydrogen, not oxygen, not nitrogen.
Do not get distracted by the biological importance of oxygen or nitrogen when you see that multiple choice question. The textual answer is carbon and hydrogen.
And fact number 5.
Fact number 5. The chemical tool used to prove this is elemental analysis, utilizing destructive techniques like combustion and ash testing. And the logical biological proof relies on testing a broad spectrum of life specifically, a plant tissue, an animal tissue, and a microbial paste against the baseline of the Earth's crust.
Those are incredibly clear anchors. To lock that in even further for everyone, let me give you a rapid-fire sticky summary.
Let's hear the 60-second version.
This is the version you can replay in your head when you are staring down a tricky, neat question.
Okay.
Imagine this scenario. You, a fern plant, a microscopic amoeba, and a jagged chunk of the Earth's crust all walk into a laboratory and step into a massive combustion analyzer.
A big machine.
The machine burns all of you down to your basic atoms, processes the data, and spits out a receipt.
Okay.
The analyzer looks at the receipt and says, well, look at this. In absolute terms, you are all made of the exact same elemental building blocks. The raw materials on this list are identical.
Right.
But then the analyzer looks a little closer at the living thing, you, the plant, and the amoeba, and it points at you and says, but you guys just decided to hoard way more carbon and hydrogen.
Hoarding carbon and hydrogen.
And that right there, that hoarding of carbon and hydrogen to build massive, intricate macromolecules driven by enzymes into a non-equilibrium steady state, that is the fundamental secret to biomolecules.
It is the chemical spark that differentiates the living forest from the dead rock. That is a perfect visualization. If you listening can remember that analyzer and that receipt, you will never get trapped by the absolute versus relative question ever again.
You'll be bulletproof.
And you will fundamentally understand the premise of everything that follows in the chapter.
I feel like we've completely reframed how to approach this topic today.
I hope so.
It's not just a boring intro paragraph. It is a massive philosophical and chemical statement about the very nature of life itself. Unpacking it like this really changes how we view the entire biology syllabus.
It stops being a list of disconnected facts to memorize.
Yes, it starts being a highly logical, mechanical story.
As it should be.
Thank you to everyone listening for joining us on this deep dive. I hope you feel as prepared and excited to tackle Chapter 9 as I do right now.
Building a solid foundation is the most important work you can do as a student, and we have laid down serious bedrock today.
We definitely have.
But before we sign off, I want to leave you with a final provocative thought.
Oh, I always love a good puzzle to end on. What is the thought?
Well, we have established today that we are made of the exact same chemical element as a lifeless rock, just arranged into more complex polymers, interacting through metabolism and sped up by enzymes.
Right.
My question for you to ponder is this. At what exact specific microscopic step in that chemical assembly line does a pile of interacting carbon and hydrogen actually become alive?
Oh, wow.
Is life something that happens precisely when the first peptide bond forms? Is it when the first enzyme lowers an activation energy?
Or is it bigger than that?
Exactly. Or is the living state an emergent property that only exists when the entire chaotic system of millions of reactions is running simultaneously?
That is heavy.
Think about where the exact boundary between a complex chemical reaction in a beaker and an actual living organism truly lies. Think about that as you dive into the rest of the chapter.
At what exact step does chemistry become biology? That is the ultimate question. Keep that in your mind as you study the macromolecules and the pathways. Until next time, keep digging deeper, keep asking why, and we will see you on the next deep dive.